Welcome to Medicines, Small Molecules, and Proteins. Please take a moment to review the learning objectives for this lesson. Let's start with defining the term medicine. The legal definition of a medicine, medication, or simply drug is a substance intended for use in the diagnosis, cure, mitigation, treatment, or prevention of disease. Many of us take medicines every day in the form of pills. The active ingredients of pills are generally known as small molecules. Let's take a closer look at small molecule medicines. As their name implies, small molecules are much smaller than a protein or mRNA, more on the scale of a single amino acid or nucleotide building block. Therefore, many can easily cross cell membranes to work inside our cells without need of a special delivery system. Small molecule medicines have multiple advantages. First, most are able to be formulated into solid forms that are stable for long times at room temperature. Secondly, most can be taken by mouth, and once dissolved by the digestive system can go everywhere in the body, including, sometimes, the brain. These small molecule medicines include antibiotics such as penicillin and painkillers such as ibuprofen and acetylsalicylic acid, or aspirin. And the list goes on and on, from medicines that treat hay fever, nausea, high blood pressure, and high cholesterol, to those that treat psychiatric disorders like depression and schizophrenia. Nonetheless, there are some noteworthy limitations. In particular, current-day development of each new small molecule medicine generally involves an initial screen through tens of thousands of compounds, followed by years of medicinal chemistry to mold an initial hit into a potential medicine. Following that, there are years of preclinical and clinical studies to prove that it is both safe and effective. Unfortunately, most small molecules drop out somewhere along the development journey, due to either unanticipated toxicities or lack of efficacy. Because of the need to screen so many compounds and the high failure rate, small molecule drug development is both very time-consuming and very expensive. The way almost all small molecule medicines work is that they bind to a particular protein and change how that protein functions. Some small molecule medicines slow down or completely inhibit the target protein's function. Others do the opposite and increase the target protein's ability to function. Therefore, modulating the activity of proteins is a general mechanism by which we can treat ailments and disease. But proteins themselves can also be medicines. They now make up a key class of medicines known as therapeutic proteins or protein biologics. Almost all proteins currently in use as medicines do their work in the bloodstream. Notable examples include insulin, used to treat diabetes, blood clotting factors used to treat hemophilia, erythropoietin used to treat anemia, and monoclonal antibodies, which have many different uses ranging from warding off viral infections to treating cancer and arthritis. As a class, monoclonal antibodies have proven so efficacious that between 2015 and 2020, the U.S. Food and Drug Administration, the FDA, approved about 10 new monoclonal antibody medicines per year. Individual proteins from viruses and bacteria can also be used as medicines, in this case to prevent future infection. These are called protein subunit vaccines. Since they only constitute one small piece of the infectious agent, protein subunit vaccines are generally safe and cannot cause disease. Instead, they prime our immune systems to recognize and respond to that specific virus or bacteria in the future. Now, let's discuss some of the disadvantages of protein medicines. Despite their advantages, protein medicines also have notable limitations. For example, for the first several decades of its use as a medicine, insulin was purified from animals. However, animal insulin has a slightly different amino acid sequence compared to human insulin, and was therefore prone to causing allergic reactions in patients. Even more dire consequences were suffered by hemophiliacs. Prior to the development of recombinant clotting factors, hemophiliacs were treated with clotting factors purified from donated human blood. Contaminated blood products led to thousands of patients contracting hepatitis in the 1960s or AIDS in the 1980s. This was only corrected when appropriate screening procedures were implemented in 1969 for hepatitis and in 1985 for HIV. While some therapeutic proteins are still purified from natural sources, these days most are made using mammalian cell lines that have been genetically engineered to manufacture the protein of interest. A word commonly used to describe these cells, as well as the therapeutic proteins they produce, is recombinant, which loosely means a new combination. Recombinant cells are grown in huge stainless steel vats called bioreactors. Each bioreactor requires significant industrial infrastructure to keep the cells alive and free from contamination. So making therapeutic proteins is a very expensive endeavor. Further, because each therapeutic protein is unique, each one separately requires significant work to figure out how best to stabilize and formulate it so that it doesn't degrade during storage and ultimately functions as intended once injected into the body. Finally, most proteins can't easily enter cells, meaning that almost all therapeutic proteins are limited to jobs either outside of cells, for example in the bloodstream, or in the special compartments known as endosomes and lysosomes that cells use to take up nutrients from their environment. But more than 90% of human proteins reside either completely inside cells or are embedded within cell membranes. So if we tried to make and use these intracellular and membrane proteins as medicines, they wouldn't be able to get to the proper location that they need to function. This is where the idea of mRNA medicines comes in. Instead of supplying the human body with a pre-made protein, we could supply just the instructions for making that protein, that is, the mRNA. This would enable our bodies to manufacture our own therapeutic proteins. And because these therapeutic proteins would be made inside our own cells, any type of protein, intracellular, membrane-bound, or secreted, could become a medicine. It sounds so simple, but of course it's not. Our ability to use mRNA as a medicine has only recently become possible due to decades of hard work by both academics and industry researchers. In the later lessons of this module, we'll learn about the many barriers that had to be overcome to enable the creation of mRNA medicines.